This is the Mycological Association of Washington. This is our August meeting. And obviously our continuing presence on the online format. And just a couple of notes about the meeting tonight. And so first of all, we had a board meeting, which we have for a year, last Thursday, and we discussed kind of the first board meeting we've had since the restriction against seeing each other with the pandemic restrictions, and kind of been working on how presence with the online format. So we're going to allow, we had before restricted to members only because we were worried about numbers, but that number one doesn't appear to be a problem. And number two, we're going to expand the numbers allowed. So the point is with our programs, and they're listed very distinctly by month on the MAW website. So I don't need necessarily to go through them here, but we have a fairly robust program. So please feel free to invite anyone who you would think might be interested to look in. And by the way, that was when we were at the library, we allowed everybody. There was no requirement when we were at the library to be a member. And so we held off on that because we were afraid of the limitations that we might have online. So we now are going to change that policy to allow anyone to attend and to try and hopefully broaden some of the things that we're going to be doing. The second thing on the board meeting we talked about was that we had been skipping January as a program, but obviously without the problems of weather. Again, one of the benefits, the silver lining of online meetings is that we don't have to worry about things like the hurricane, whatever they named it. They're running out of names, obviously. It was Isaias or something. I couldn't even quite pronounce it, nothing. But anyway, so we're going to continue to have meetings every week, or every month, rather. The other thing we're going to try and do virtually, in addition to meetings, is some sort of 4A activity. Mitch will talk about that when he does the fungal review later in the program. He's the 4A chair. And also, we are looking at trying to do some sort of an online cooking demonstration or cooking participation, and something like do a recipe separately and then get together, or show cooking or some. So that's under review right now for potential future activity. So the last item that is of general interest is every year we have an election, as you may have experienced that in the past. And since this is sort of a special year, we're starting early because of normally we have more activities like the mushroom fair, sequinota, etc., where we'd be able to talk and meet with people. But so the election is not until the end of the year. But we have nominated, and the way it works is you have one board member and then two non-board members form the nominating committee. So the board member is John Harper, who is the culinary chair. He is going to be and he will be looking for two non-board members who would be interested in putting the slate together, or the list of candidates. And secondly, if you are interested in joining the board, that's the time to let that be known also. That'll come out. Our nominations officially will be in November. So lastly, if you need to contact John, for example, or any of us, I always say this, it's the title and then at MAWDC.org is the email. So I would be president@MAWDC. And if you forget that, it's on the website, and I'm sure you know where that is. And under About Us, there's a whole list. There's a list of the members of the board members and their email addresses. So it's pretty easy to find that. And finally, I just mentioned planning. We are already looking to next year, so we already do have the mushroom fair date set, because actually we're canceling everything pretty much other than these activities this year online. But hopefully by next October 17th, which is when we currently have the mushroom fair scheduled at Brookside Gardens. So are there any other input, general input that any other board member would like to make? So then what we have done in the past is we have two kind of one mini program and one main program. The smaller program will be on the subject of slime molds. And I'm going to turn this over now to Lamont Yarroll, who is I got Lamont part right. It's Yarroll, who's from Western Pennsylvania Mushroom Club. We're just going to talk about slime molds for roughly 20, 30 minutes. So unless there are any other input from the board, I'm going to turn it over now to Lamont, who is going to introduce himself more fully and then start into his presentation. So Lamont, you have the floor.
Thank you very much. All right. Whoops. Okay. So this is just a little taster for slime molds. I don't go into a lot of depth, but I'm hoping to convey some of my enthusiasm for these interesting creatures. So thank you very much for hosting me. All right, so something you might not know, slime molds are in fact not fungi. It's kind of a historical accident that the people that study them are mycologists. They produce spores, but that's about the limit of their similarity. They're protozoa, and here we have a lovely example of a streaming plasmodium. This organism is climbing up the rock looking for lots of bacteria to eat. They use a protein quite similar to the one that we use in our muscles. So let's start off with one that you've probably seen a lot. As you can see, when you're walking along, it doesn't look very dramatic. You just kind of have these little white things on a log. But in fact, when you get up close, they're really quite beautiful. This is the Paroides variety of this slime mold. Now, all other slime molds produce a fruiting body that contains the spores, but the Ceratiomyxas have all their spores on the outside. And even if you look closely at the photograph on the right, you'll see that they look a little fuzzy. Those are the stalks that are holding the spores. So this one's fun to find. It's very difficult to collect because it's super delicate. Kind of turns to water as soon as it gets touched. So, okay, and here's another one you've probably seen, most likely in your mulch, Fuligo septica, also called dog vomit or scrambled egg slime mold. This is one of the few slime molds that I actually eat. Tastes like broccoli, really good broccoli, in fact, when it's fresh. And again, we can see the smaller photo on the right here. Does my cursor show up? Yes? Okay, good. We can see that this colony was actually moving when it decided to turn into a permanent fruiting body. This is not the condition you want them in when you're going to eat them. You want them more like this, soft and yellow. And there is a variant of this, Fuligo candida, the white dog vomit slime mold, which tastes like cucumber, so very strongly cucumber. And there are two lookalikes, neither of which taste like cucumber. Brefeldia maxima, the Tapioca slime mold, and Mustelago. So they're safe to taste. I wouldn't eat large quantities of either of these, as I don't know what deeper edibility there is. Now, this one is just plain beautiful. If you get the opportunity to see it, they can get largish. I've seen them a couple centimeters across. Here's an unidentified tubulifera on the right here that fruited repeatedly. These are Really nice, colorful, and they really stand out. Okay, and these Hemitrichias can be difficult to spot, even though they're bright red to bright orange. These photos are both pretty close up. Those are maybe 1 to 2 millimeters across. They look like little red lollipops. And the reason both of these are labeled Hemitrichia species is they're immature. Actually, identifying which species of Hemitrichia it is requires them to be fully mature. And here's some lovely photos from George Barron of mature Hemitrichias. So it turns from that bright red lollipop to this yellow fuzz. And Caliculata has this little chalice at the bottom. Clavata is club-shaped. The other notable thing about them, clavata has a single point of attachment inside that cup, and Caliculata is connected all the way around. Those are the two species of Hemitrichia that we find pretty commonly. I wanted to show you a Hemitrichia photo micrograph, but I couldn't find one. So here's one of Trichia, which is the genus that the Hemitrichia was originally put in, but then they were split out into a separate genus. So this is botrytis. But you see these double spirals? They're really beautiful. They're called elaters, and they throw the spores out as they uncurl. Really quite beautiful under the microscope. This is another one you may have seen. You get these, they vary from bright red to kind of this brick red. Linkoff calls it the cotton candy slime mold. These are really lovely, both with a magnifying glass and under the microscope. So here's what they look like under the microscope. You have these little plates all along the elaters, and the elaters come together and form a net rather than just single pieces. So, all right. And here is another really lovely one. I don't find this one very often, but their cancellata means railing. So it's like ironwork. So here's a close-up photo. You can see the cross connects between the ribs. This one's, I really like finding this one. And here's another Cribraria. I don't have a photograph of this one at full size. This is just this close-up, but you have these lovely nodules that form the net. So this is a curve. This photo covers maybe 3/4 of a millimeter. So, okay, another popular one of the Stemonitis species, the chocolate tube slime molds. I have gotten very hesitant to identify these in the field. A couple of books I've found have claimed that the ratio between the stem and the upper part of the fruiting body can be used to differentiate species. And I found that really unreliable, that you really need to get them under the scope. So when they start out, they're actually white, and you start seeing this kind of chocolate icing appearance. First as just icing, and then at later date chocolate icing. Then they kind of break apart into these fuzzy hair things. And then here's a close-up of one of those So just one of those tubes. And this is what you need to look at in order to differentiate the species. So you care about this outer net. You want to look for ends to see if there are ends sticking out or if they're all closed, like this one. And the stem down the middle, called a columella, is connected to the outer net, in this case, by relatively vertical stanchions. They support the net directly. Sometimes there's a second net on the interior. Sometimes you have to look at the end of the columella to see whether it goes all the way to the end or if it kind of breaks up into branches. So, okay, so here are 3 references that I recommend very highly. Some of the photos were from George Barron, those Hemitrichias, so that those are from his website that goes with this book. And the reference for this, Le Myxomycetes, is an excellent photographic reference. They have 2 volumes. One volume is photographs and drawings of microscopic structures, and the other volume is the keys. And the keys are in French and then again in English. So I was able to pick these up for maybe $110 for the 2-volume set. It's a pretty good deal if you're interested in Myxomycetes. And then Stephen Stevenson is probably North America's premier myxomycetologist. He has a nice little book that's very approachable, and he gives more about the ecology of Myxomycetes and the roles they play, and also gives some developmental background, differentiating the plasmodial stage where they move around and the fruiting stages. So I can recommend all of these books. And I'll slip one more in. Fungi of Temperate Europe. This one just arrived. They have a nice large section on Myxomycetes. And Thomas Lasso and Jens Peterson. They've co-authored a number of good mushroom books. These are as excellent as any. Well, thank you very much.
The chat lit up when you talked about being able to eat slime molds. It's kind of predicted. So can you tell us more about how you would actually go about that? And is there a source you would recommend that specifically has information about, you know, those species and how you do it, and how careful you need to be, whether other ones are poisonous, those sorts of things?
Right. There's very little edibility data on the slime molds. Fuligo septica we know is edible because it's farmed commercially. So in Mexico, it's called Luna de Caca. Caca de Luna, sorry. Caca de Luna. So moon poo. I'm told that it's generally cooked. Then it develops a taste more like peanuts after being cooked. But I snack on it raw.
Wow.
Yeah, and you get that really good broccoli flavor. So it's good mainly for shock value in a walk. It's pretty rare for me to find enough to call it a meal. So, and most of the other slime molds don't produce large fruiting bodies that you would even think about eating. You know, where These things are millimeters long. So just the prospect of gathering enough of them to eat is bizarre.
Yep. We got one question from someone about what's the best way to photograph? I assume sort of, so small and,
Yeah. I've been trying to improve my photographic technique. I have a decent point and shoot that has a very nice macro mode. But even at that, you need good lighting and just to get these photos. Let's see, these, right? I'm pretty sure he's actually doing focus stacking, where you're taking a whole bunch of photos at different focal depths and then composing them in order to get the full depth of field. The other possibility is these are shot with really good lighting also. I don't think this was using photo stacking, but yeah, so I'm still learning. They're a challenge to photograph. My latest phone has a macro mode, a separate macro camera, and I'm hoping to get good use out of that. And I also have some clip-on lenses for cell phone that allow macro mode. So get up close, get it as much light as you can. Uniform light is important. If you're out in the sun, you may actually want to shadow it with your body to make the lighting more uniform.
Can you talk just a little bit about how slime molds move during the plasmodial stage?
Yeah, so they're famous for being able to solve mazes. And essentially what they do is they extend pseudopods by applying pressure, and then they pump fluid from one section of the plasmodium to another along tubules that they produce. And it's really quite remarkable. But it's the same technique that an amoeba uses to move. So they're because I mean they're very close relatives of the amoebas.
And the last question, oh, there's a couple more in the chat. One person said, does the fruiting body contain the protozoan?
No, the protozoa will come together, and in fact, generally they don't have cell walls. So you can either say it's a colony of cells with no walls, or you can say it's a multinucleate cell. And it's still not fully understood how they decide what cells become what parts of the fruiting body. But it is definitely a whole bunch of the plasmodia that come together to form each fruiting body. So I wish I had a better answer, but that's what I know.
And around here, we often see on oyster mushrooms, there's a yellow, I assume it's a slime mold.
Oh, on the oyster mushroom.
Yeah.
I believe that's a, well, there is a slime mold that likes some of the guild fungi. Most of them eat bacteria, but there are a couple that'll eat fungi. But there's also a, oh, come on, one of the Sordaromycetes. I keep thinking Hippocreos.
Fusarium polycephalum.
Oh, polycephalum, yeah, that's the slime mold. In fact, yeah, this is probably polycephalum, though it's too young to tell for sure. There are a lot of slime molds that produce a yellow plasmodium like this. So, okay, so.
So Rick's asking, how do they reproduce? You showed us the spores.
Mm-hmm.
I'm not sure what else. You want
Yeah,
to know,
they're
but-
it's sexual reproduction. Let's see. Get some spores. Yeah, here's some spores. Oh, actually, the background is spores too. So these individual spores will typically have a plus or minus designation, roughly equivalent to gender. And it's a mating compatibility level. And then they will form a, they can either form a plasmodium from a single spore, in which case it's a haploid plasmodium with just the one set of genes, or they can fuse and become a diploid plasmodium. So actually surprisingly standard sexual reproduction.
So our monthly talk last month was on mushroom reproduction, and it was much more complicated.
Yeah, there are some. Yeah, I would say the slime molds on the whole are relatively simple in their reproductive gene swapping strategies.
Cool, cool. All right. And for anyone that missed that meeting last month, we've actually got them up on YouTube now. There's a MAWDC channel on YouTube. And I think Tom sent around the link in one of his last emails, but we'll send out the link to this one when it's up too, so you can see them.
All right. And I'll make the slides available to Ma.
Awesome. Thank you so much.
Sure. All right. I can't seem to stop sharing.
I think I can stop it.
Okay.
There you go. All right, back to William.
Okay, so thank you very much, Lamont. A couple of brief notes on our typical fungi in the news. Just two brief notes I thought quite interesting. I find these things, this is from The Economist, and they're very clever about their names. It's called Champignon Champions. It's about the province of Yunnan in China, which they claim have, the country has 1,000 edible mushrooms, of which Yunnan has 800. And the article is about that. And they have one mushroom that they call the goat belly that sells for 1,000 yuan per pound or per kilogram. And they call the morel the goat belly. So I did so that's about $70 per pound roughly. So and they claim they find several new types of edibles. So they must do an awful lot of trying things that don't work. The other mushroom that's mentioned is called the little white, which apparently is like our little white, the destroying angel, only it looks more like an oyster, and people die from eating that one. So I guess, anyway, I did not realize there was that broad a mushroom, variety of edibles in China. It's an interesting article. The last thing I wanted to just mention is Fungi Magazine, which if you don't get it, it's a pretty good thing to have on your bookshelf. The last issue, which this one, is really got one section quite fascinating. It's almost a Plato-Socrates discussion about the types of mycorrhiza, which I'm sure you're familiar with mycorrhiza. If you're not, it's this fundamental. Why I got interested in fungi was the fact that 90% of all plants require them, which you probably know. But this talks about the difference between the two basic types, endomycorrhizal and ectomycorrhizal, what they bring to the different, and the fact that there are 110 genera of plants that have both. And so this is one goes inside the cell and one is around the cell. We had always more or less thought that they were discussed them as separate. Trees were ectomycorrhizal, which means surrounding with the Hardig net. And then The rest of them were what's called endomycorrhizal, with vascular or vesicles and arbuscules inside the cell. But this contends that no, that's not the way it is, and it makes a lot of sense. It's very good reading, and it kind of opened my eyes to a whole new aspect of the mycorrhizal relationship. So that's what's in the news. So I'm now going to turn it over to our 4A chair, Mitch Fournay, who's going to take us through a list of things that people have sent in to him over the last several weeks and discuss them as he would do at the front of our meeting normally at the mushroom table. So here is Mitch and the mushroom table.